Hi everyone, continuing our discussion from the last time, we had derived expressions for T1 and T2 for a bacterial population evolving in an environment where population size is held to be constant equal to K. T1 was associated with the time that I have to wait for the first beneficial mutation which does not get lost due to drift and gets established in the population. Once this beneficial mutation is established in the population, selection takes over and its numbers go up to n, and the mutation is said to be fixed in the population. T2 is the time that this beneficial mutation takes after getting established to reach fixation. And in the last lecture, we had obtained expressions which accounted for this T1 and T2 in terms of number of generations that I have to wait for each of these two events. And now let us continue our discussion from there. So, this is the expression that we had received for T1 and this is what we had got for T2. And as we had discussed before, that once a beneficial mutation is established and fixed in the population, fitness of every single individual in the population has increased from 1 to 1 plus S. Starting genotype, every individual was at fitness 1, and now since the first beneficial mutation has become fixed in the population, fitness of every single individual is equal to 1 plus S. That means that mean change in the fitness that has happened is equal to S, and that change S has happened in time T1 plus T2. So, speed of evolution can be written as S divided by NUS plus 1 upon S ln of NS. Let us call this speed of evolution equal to V, and that is equal to fitness per generation. So, that is the first result that we have in which we can quantify the speed of evolution in an analytical manner and come up with an expression that defines how fast is a bacterial species evolving. But let us now take a closer look at how does this experiment actually play out in a physiological setting where this experiment is actually being conducted, and try and understand some of the implicit assumptions that have gone into development of this expression. So, the way we have developed this system is, let's just take a step back and think about what we have assumed here. The way we have developed this expression is that we have an environment E where there are n individuals and one beneficial mutation occurs in this environment. This beneficial mutation might get lost or become fixed. Suppose it becomes fixed, then this mutation is acted upon by selection and its numbers, the number of individuals corresponding to this particular mutant genotype, quickly increases to n. Then we have to wait for the next beneficial mutation to become fixed in the population, and so on and so forth. The implicit assumption that we are making in this whole analysis is that at any particular instant in the system, there is only a maximum of two genotypes that coexist, the parent genotype and the mutant genotype, because once a mutant genotype arises, its numbers increase and go towards n. So, the implicit assumption in this model that we are making is that this is time again. The implicit assumption that we are making is that I am not going to draw the mutations that were eliminated by drift, but only the ones that get established. So, and then the next one occurs somewhere over here, and so on and so forth. The implicit assumption is that if I were to draw a vertical line which corresponds to a particular time, so this is like a snapshot of the environment at each of these times. In any of these snapshots, the maximum number of genotypes that exist in the population is equal to 2. So here every individual belongs to the parent genotype, so there is only one genotype, again one genotype. Here there are two genotypes with this many number of mutant individuals and this many number of the parent genotype. So this is 2, 1, and again mutant genotypes, the parent genotype, so this is 2. So the maximum number of genetic variability that exists in the population is limited to 2. We do not account for the fact that it's plausible that while this mutation was becoming fixed and was becoming fixed after establishment, there could have been another mutation that also got established and was going towards fixation. And if that was the case, then we have a scenario where at this particular instant we have this mutant, some individuals which are this new mutant, this is the number of the first mutant, and still some original parent genotype left in the population, and the genotypic variability is 3 distinct genotypes. We don't account for the fact. We don't account for the fact that the number of unique genotypes that are coexisting in the environment at any particular time could go beyond 2. That assumption is valid if the T1 expression that we have derived is much greater than T2. Remember, T1 is the time that I have to wait for the mutation to get established. T2 is the time that I have to wait for the mutation to get fixed once it's established. What this is telling us is that if on average T1 is much bigger than T2, that means that the time that I have to wait for the first mutation to get established is much longer as compared to the time it takes for the mutation to go from establishment to fixation. Then the assumption that I only see a maximum of 2 distinct genotypes is fair, because since the waiting time for a mutation is very, very large, the chances that 2 mutations of beneficial nature will arise and get established in the population next to each other are going to be very, very small. Because one beneficial mutation becoming established from here, then the waiting time for the next beneficial mutation to get established is very large compared to the time that this mutation would take to go to fixation. Hence, the implicit assumption in all of our analysis is that T1 is much larger than T2. So what we'll do before we proceed is check this assumption and find out the physiological conditions under which this particular assumption holds. So again, all of our analysis, what we have discovered holds if T1 is much bigger than T2. So what are the constraints on the variables of our system which ensure that this is true? So once again, let's rewrite our expressions for T1 and T2, and we get 1 upon NUS should be much bigger than 1 upon S ln of NS. We can cancel the S's from the two sides, and the condition that we get is 1 upon Nu is much bigger than ln of NS for our analysis to be valid. Now what does this tell? Let's try to think about this a little more. What are these variables? N is the population size associated with the environment that I'm dealing with. U is the beneficial mutation rate associated with the bacterium, and S is the selection coefficient associated with each beneficial mutation that is occurring in the system. So let's try and get realistic estimates of S and U, the selection coefficient associated with each beneficial mutation and the beneficial mutation rate that's happening in this bacterium, and try to get estimates of N such that this relationship holds. That will give me an idea of what is the size of the environment that I'm looking at in terms of number of individuals that environment should support, such that this assumption holds and all of the previous analysis that we have just completed is also valid for this environment. So what would be a good representative value for S? Large beneficial mutations we know confer an advantage of 4 to 5%, but those beneficial mutations are relatively rare. So, S of perhaps 2% is a realistic representation of an average benefit that a beneficial mutation confers to an individual. So, S, let us take that as 0.02. U is the beneficial mutation rate. The total mutation rate of E. coli is 10 power minus 3 mutations per cell per generation. I am sorry, this is 10 power minus 3 mutations per cell per generation. Now, we understand that this mutation rate encompasses all mutations, deleterious, lethal, neutral and beneficial. So, the beneficial mutation rate is bound to be lower than this, and if we imagine that 1 in 100 mutations is beneficial in nature, that means this mutation rate can be estimated as 10 to the power minus 5 mutations per cell per generation, which are beneficial in nature. Now, once we have these two numbers, I leave this as an exercise for you to come up to solve this inequality and come up with an estimate n such that having plugged in these values of S and U, the above inequality holds. The above relationship holds. Okay. So, you should try and solve this and find out that what is the constraint on n that you are putting when you are saying that this inequality must be obeyed. What is the constraint on n that automatically imposes itself. So, solve for n and comment on what is the nature of the environment E, where you can do this experiment with this constraint on n such that T1 is much bigger than T2, which means that a picture like this that we drew, where one beneficial mutation occurs, goes to fixation, this one does not exist, then we wait a long time before the next beneficial mutation gets established. This picture holds true depending on what is the answer that we get from this inequality. Okay. So, that is that. But what that also means is that if T1 is much bigger than T2, then we can write that if T1 is much bigger than T2, then we can write that T1 plus T2 is approximately equal to T1, which just means that the rate of evolution is just equal to S upon T1, which is S upon 1 divided by NUS, which is equal to NU S square. Again, this relationship is valid in a regime where mutations are rare. Which is essentially what saying T1 much bigger than T2 means, because T1 is the waiting time that you have to wait before the first beneficial mutation gets established. If T1 is very large compared to other times in the process, that means the waiting time is the determining factor which decides speed of evolution, and this will only happen. Another way of saying the same thing is that mutations in the system are very rare. So, that is the speed of evolution associated with the condition that mutations are very rare. Another way to think about this is that if we try, our goal is to try and estimate an expression for speed of evolution. So, another simple way to think about this is speed of evolution is just equal to, if I were to ask you that in one generation time, how many beneficial mutations occur? So, we are going to write this as how many beneficial mutations occur, not fixed but occur in one generation. Okay. We need an expression for this, times what fraction of those get established, we want an expression for that, times what is the fitness advantage of each of these mutations. So, what you are saying is that in one generation how many mutations occurred times what fraction of those got fixed. So, this gives you the total number of mutations that got established in one generation time. So, if this number is, let us say this number of mutations that occurred is 100, then each gets established by a probability of 0.1. That means the total number of mutations that get established in one generation is equal to 100 times 0.1, which is 10. And if each confers a fitness advantage of 2%, 0.02, then you have 10 into 0.02, 0.2 as the 0.2 fitness increase per generation as the speed of evolution. So, if we want an expression for each of these quantities, so number of mutations that occur, and again when we say mutations, we are only talking of beneficial mutations here. So, the number of mutations that occur is equal to NU. The fraction of these that get established is going to be equal to S, because we already know that a beneficial mutation gets established with the probability equal to its selection coefficient. This is S, and each of these beneficial mutations confers a fitness advantage equal to S, as we have assumed in our model that all beneficial mutations are equivalent in nature and each confers a selection coefficient S, and that is the benefit that this mutation upon fixation confers to every individual that is present in the environment. So, this quantity will again be S. And this just gives me speed of evolution is Nu S square fitness per generation, which is V. Okay. Which is what we had obtained in the last slide, when we had assumed that T1 much bigger than T2, and hence T1 plus T2 is approximately equal to T1, and we substituted rate of evolution as mean change in fitness divided by the total time in which that change in fitness has taken place, which is equal to T1 plus T2, but under these conditions can be approximated to be just equal to T1, and hence you get the same expression from either of these two ways to think about this. Okay. So, this was analysis considering that mutations are rare. Beneficial mutations, you have to wait a long time before a beneficial mutation occurs and gets established in the population. But in a physiological setting, if you work out the NU and S inequality that we had developed and find the appropriate condition where this inequality, that waiting time is very large compared to fixation time holds, you will find that this scenario is not really relevant for a large number of physiological settings, because NU, that is the total number of mutations that are occurring in one generation time for any bacterial system, and because N associated with the system is likely to be very large, NU is often a very large number, that is a total number of beneficial mutations happening in this system. If NU is large, that means mutations are not rare and waiting times are not as large as compared to what was going on in the picture that we had just drawn. So, mutations are actually fairly rapid, and what we are going to do next is derive an expression for speed of evolution in a case where mutations are happening rapidly. But before we try and develop that expression, let us try and intuitively understand what would the picture look like in a setting like that. And to understand that better, we go to one of the other representations of output from an evolutionary experiment, because I think that gives a better insight into what is happening in the system. So, this is a case where mutations are occurring rapidly now. Before we do that, it's perhaps useful to understand what would be the corresponding picture in this representation that we are going to talk about now, and understand what would this look like when the mutations were rare. So, this is representing what we have already done. Mutations are rare, and in this representation y axis is number of cells, this is n, and x axis is fitness. So, in what we just did, when mutations are rare, you have all individuals at fitness f naught. This is at t equal to 0. Then at some point you have one beneficial mutation occur. So, this is at t equal to 0, all individuals at fitness f naught. As we go forward in time, one beneficial mutation occurs and that gets established. I am not labeling the axis, but the same axis labels carry here as well. So, at this time now, one beneficial mutation has occurred and you have n minus 1 individuals of the parent genotype with fitness f naught, and you have one individual with fitness f naught plus s. This is 1. This is t equal to t1. Now, this mutation, let us imagine that it does not get eliminated due to drift and is able to establish itself. If that is the case, then at t equal to t2, what the picture is going to look like is that the number of individuals corresponding to the parent genotype at fitness f naught is going to be n minus 1 by s, and this one mutant has increased in numbers and gone up to 1 by s number of individuals which correspond to this particular genotype. And now the mutation has gotten established. This is t equal to t2. Now selection takes over and fixation of this mutation happens. And fixation of mutation happening means that this particular genotype is going to outcompete the parent genotype, and eventually you will end up with all individuals at f naught plus s and 0 individuals at the original fitness f naught. This happens at t equal to t3. Then under this regime that we just discussed, fitness mutations are relatively rare. You have to wait a long time before the next mutation occurs at t equal to t4 and gets established. And in that case, at t equal to t4 now, you have n minus 1 individuals at f naught plus s, and you have one mutant individual that has arisen, which is f naught plus 2s. And the whole story repeats itself, and this is equal to t equal to t4. And the idea being that once the population reaches t equal to t3, it takes a long time for the system for the first mutation thereafter to get established, and t4 is much larger as compared to the other time steps associated in this process. So, if this is the setting that we have for rare mutations, in an equivalent setting, what do you imagine is going to be happening when mutations are no longer rare but are much more rapid in nature? So, NU is a very large number, which is the total number of beneficial mutations occurring per generation. NUS is the total number of beneficial mutations which are going to get established per generation. If this number is much larger than 1, then what you have is that more than one mutations are getting established per generation. So, if we draw the equivalent picture in this regime when mutations are rapid, what would that look like? So, now we are interested in a regime where mutations are rapid. What is this going to look like? The way it's going to play out is the following, that we have fitness here, F0, F0 plus S, F0 plus 2S and so on and so forth, and we have number of cells on the y-axis. And to start with, if this is n, there were n number of individuals at fitness F0. And as I go forward in time, the cells are growing. Eventually what is going to happen is that mutations are going to keep on happening, and some of them will be lost because of drift, but one mutation will eventually get established. When that happens, number of individuals which correspond to fitness F0 will come down, and you will have some mutation, some number of individuals which correspond to a fitness of F0 plus S. And now because this mutation has gotten established, the numbers associated with this particular genotype will increase, numbers associated with the parent genotype will decrease. But before this can reach fixation, that is before this genotype can totally eliminate the parent genotype from the environment, another mutation from this pool happens, and you have another genotype which corresponds to fitness F0 plus 2S. This event has meant that now you have three distinct genotypes in the population at the same time, and we had precisely seen that mutations are rare implies that there are not more than two genotypes at any given time in the environment. So, we have three. So, now this particular genotype is going to outcompete both the genotypes. The frequency associated with the middle genotype will keep on increasing as long as this fitness F0 plus S is more than the mean fitness of the individuals in the environment. So, if the frequencies were as represented, this will go up, this will go up, this will come down. But before this individual has gone to fixation or this has been completely removed from the system, another mutation happens and we have 4 genotypes. So, what mutations are rapid? This regime implies that multiple genotypes, and by multiple it is greater than 2 genotypes, coexist at the same time. Because we are not limiting beneficial mutations occurring in the environment, beneficial mutations are constantly available to individuals of the environment, the fitness will keep on increasing. What we are interested in is that what shape does this graph take when the system takes on, when we wait for a very long time and observe this graph take a particular shape. What is that shape going to be when we have waited for a very long time and the system has had time to accumulate accumulations? And that's something that we'll start the next lecture with. Thank you.